When the human heart stops beating, a remarkable and complex transformation begins, one that goes far beyond the instantaneous cessation of life. This first minute after the heartbeat fades is a key stage during which the transition from a living system to a lifeless one can be observed. Pathologist Artem, who has devoted many years to studying these processes, explains that, for him, death is not a tragedy, but a wondrous physiological dance of molecules and cells. In the first seconds after cardiac arrest, the oxygen supply to the brain is instantly cut off, and this organ, uniquely sensitive to energy deficiency, begins to show the first signs of suffering. Adenosine triphosphate molecules, the main energy carriers, cease to be synthesized, leading to the shutdown of vital ion pumps in cell membranes. Neurons, deprived of energy, begin to accumulate calcium, triggering a cascade of destructive enzymatic reactions that damage cellular structure. It's important to understand that consciousness fades approximately 6 to 10 seconds after oxygen supply is cut off, while the body is not yet completely dead. It is at this stage that clinical death manifests itself, when the electrocardiogram registers a flat line. But brain tissue still retains some residual activity. Muscles relax, breathing slows and becomes shallow, and the eyes may roll back in the head, as if reflecting the sudden loss of vital light. Over time, starting around half a minute, it becomes clear that neurons are entering a state of chaos. They release glutamate, a powerful neurotransmitter that, in excess, causes toxic excitation. This causes neurons to spontaneously fire, resembling a chaotic fireworks display in the brain, like the last flashes of life before the final darkness sets in. This excitotoxicity accelerates damage, causing apoptosis and cell necrosis, with features particularly pronounced in the hippocampus and cerebral cortex. After 3 to 4 minutes of airlessness, an irreversible process of cellular depletion begins. Membranes are destroyed, mitochondria disintegrate, and enzymes, released from lysosomes, destroy cells from the inside. At this point, edema begins to form in the surrounding tissue, caused by the outflow of ions and water, and free radicals, produced in excess, damage DNA and proteins. It was during this period that hypotheses emerged about why people who had experienced clinical death report the tunnel light phenomenon, since cells in the visual cortex are among the first to die. After 10 minutes of complete oxygen deprivation, the biological stage of brain death, defined as brain death, occurs. Electrical activity on the electroencephalogram, EEG, disappears irreversibly, which is the legal criterion for death in most countries. At the microscopic level, neurons transform into so-called ghost cells, cells with destroyed nuclei, vacuoles, and loss of structural integrity. The brain becomes edematous. Intercellular fluid fills the tissues, and organ function is completely lost. Simultaneously, a complex, dramatic process begins to unfold within the body. Blood circulation ceases, and tissues are deprived of essential nutrients and oxygen. Blood begins to accumulate in the lower parts of the body under the force of gravity, forming so-called livid spots with a characteristic purple hue. A slow but inexorable cellular collapse begins. Cells switch to anaerobic metabolism, producing little energy and accumulating waste products such as lactic acid, causing intracellular acidosis. Lysosomal autolysis begins within cells, where enzymes previously isolated by membranes are released and begin to digest their own proteins, fats, and nucleic acids. This process is especially evident in organs with high enzyme levels, the liver, pancreas, and brain. These changes may be invisible externally, but upon dissection, The disrupted cellular structure becomes apparent within a few hours. At the tissue level, the first signs of disintegration appear. Epithelial cells lose contact with the basement membrane and begin to detach. Muscle fibers lose their ability to fire, entering the primary rigor mortis phase. Blood leaks from the vessels into the surrounding tissue, causing edema. In brain tissue, the neurons most sensitive to hypoxia are the first to die, as confirmed by histological examination after just 30 to 40 minutes. At the same time, active microbial expansion begins, especially in the gastrointestinal tract, where the bulk of the microflora is concentrated. Immediately after death, the immune system loses control, and microbes begin to migrate from the intestines to neighboring organs, including the liver. There, they cause tissue destruction, accompanied by the release of gases and the characteristic odor, familiar to everyone who has heard of decomposition. Alisa, a forensic microbiology expert, emphasizes that the intestines become a veritable frontline in the microbial war. Bacteria of the genus Clostridium and Bacteroides actively ferment proteins and fats, producing toxic gases such as hydrogen sulfide and ammonia. These gases accumulate and distend the tissues, causing bloating, swelling of the face and neck, and the formation of pronounced cadaveric changes visible to the naked eye. As the body cools and the immune system ceases to function, conditions become ideal for insect invasion and bacterial enzymes. The first flies lay eggs in the body's natural openings, and the larvae rapidly develop, creating tunnels and destroying the remaining soft tissue. This period marks the transition from chemical decomposition to the transformation of the body into a mushy mass with a characteristic odor and external changes. Thus, the first minutes, hours, and days after cardiac arrest are more than just a straight line on a monitor. They are a time of incredible activity at the cellular and molecular level, when life rapidly ebbs and death takes its toll, unleashing a chain of biochemical processes that destroy the body's structure. This detailed picture explains why medicine and forensic science devote so much attention to this short but crucial period. After all, it is during these moments that the body's ability to restore function is determined, or the irreversible path to death begins. Understanding these processes not only reveals the biological essence of death, but also aids in the practical work of pathologists and resuscitators. With this knowledge, we are ready to move on to the second part of this revealing journey through death, where we will examine the further transformation of the body and the influence of external factors on the decomposition process. Microbial expansion and deep phases of body decomposition. A few hours after cardiac arrest, the body gradually begins to transform into a microbial battlefield. The intestines become the key center of this expansion, as they harbor a vast number of bacteria that, at the moment of death, are no longer controlled by the immune system. The gastrointestinal tract harbors approximately 100 trillion microbes, the most important of which are Escherichia coli, Bacteroides fragilis, and several species of Clostridium. During life, the immune system keeps them within limits, preventing them from going beyond them. But with the onset of death, this boundary collapses. After a few hours, the immune system ceases to function, and bacteria begin to penetrate beyond the intestinal wall. They quickly migrate to the submucosa, then through the mesenteric venous capillaries to the liver. By the 6th to 12th hour after death, active bacterial enzymatic destruction of the liver and gallbladder begins, causing the appearance of greenish-yellow spots on the skin in the right hypochondrium. Bile, penetrating into the tissues of the abdominal cavity, gives them a specific color and promotes the development of the putrefactive process. Clostridium bacteria, which are highly putrefactive, release hydrogen sulfide, ammonia, methane, and carbon dioxide during their life cycle. These gases accumulate in body cavities, causing abdominal distension, swelling of the face and neck, and bulging of the eyeballs. The pressure inside the body becomes so high that fluid and gases are forced out through natural openings, contributing to further tissue destruction and changes in the body's appearance. Characteristic spots and coverings appear above the skin, signaling the onset of deep decay. During the first 24 hours after death, microbial expansion and enzymatic degradation intensify. At the microscopic level, tissue structures begin to change. The intercellular matrix is destroyed. The bonds between cells weaken, and the vessel walls become porous and brittle. Cells lose their osmotic balance, swell, and begin to disintegrate, creating conditions for the formation of soft, loose, and often gelatinous tissues. This process is especially noticeable in organs with high enzymatic activity: the liver, pancreas, and intestines. Parallel to microbial decomposition, a secondary process, insect infestation, begins. Flies, primarily species such as Lucilia sericata and Calliphora vicina, find natural body openings and lay their eggs. The larvae emerge within 8 to 24 hours and begin feeding intensively on soft tissue. Within a few days, they destroy up to 60% of the soft tissue within their reach. The number of larvae can reach hundreds, and together they are capable of completely clearing the body of soft tissue in 2 weeks. Microbial and insect infestation accelerates decomposition, leading to the characteristic signs of putrefaction. As tissue decays, body weight decreases, sometimes by 20 to 50%, depending on the ambient temperature and humidity. The body begins to emit foul-smelling gases, which intensifies discomfort and attracts new organisms that contribute to decomposition. At the same time, the color and structure of the skin changes. Greenish yellow and purple spots appear. The skin peels off in places, and the body takes on a blurry appearance. A warm and humid climate promotes rapid and intense decomposition, causing the skin to become loose and internal organs to turn into a pulp. In a dry and hot climate, this process slows down, and the body undergoes natural preservative mummification. The skin gradually dries out, becoming dense and elastic, resembling parchment. These differences significantly influence the rate and nature of changes in the corpse. The second headquarters of microbial activity after the gut are enzyme-rich tissues such as the liver, pancreas, and stomach. Here, enzymes and microbes work synergistically, causing softening and breakdown of structures. The accumulated waste products of bacteria and enzymes create an environment that increases acidity and alters the pH within tissues. This in turn contributes to rapid metabolic collapse and further cellular destruction. Mucous membranes and epithelial tissue begin to peel away from the basement membrane, losing their intercellular connections. Muscle fibers lose their ability to contract and enter rigor mortis, causing tissue hardening. Blood plasma penetrates the intercellular spaces, causing edema and inflammatory reactions, which, however, are part of the post-mortem process rather than a living response. Insects adapted to decomposition form distinctive channels and tunnels in tissue, facilitating microbial access to the deeper layers. Their activity accelerates decomposition and facilitates the spread of putrefactive agents throughout the body. After completing their life cycle, the insects leave behind cavities filled with putrefactive fluid and tissue debris, the scale of which astonishes even experienced pathologists. Beginning in the second week after death, a gradual transition to deeper tissue destruction begins. The intercellular matrix, exposed to enzymes and acids, loses its strength, and collagen proteins decompose, causing tissue to lose shape and density. Connective tissue in the intestines, liver, and pancreas is particularly rapidly destroyed due to their high enzymatic activity. These changes become critical for maintaining the integrity of the body. Microbial invasion affects not only internal organs, but also the soft tissues of the face, perineum, and extremities. The decay products produced by bacteria and insects alter tissue pH, increasing acidity and accelerating the degradation of proteins and fats. Significant changes in body volume and contours are observed, complicating the identification and study of post-mortem changes without specialized techniques. The environment plays a decisive role in the rate at which these processes occur. If the body is kept in a room with controlled temperature and humidity, decomposition may proceed more slowly. Outdoors, especially at high temperatures and high humidity, the processes proceed significantly faster. Understanding these factors is critical in forensic practice, allowing for more accurate determination of the time of death and circumstances. Simultaneously with biochemical processes, conditions for secondary decomposition develop, manifested by changes in tissue color, the appearance of characteristic stains, and the formation of cadaveric fluids. The skin takes on a marbled pattern, alternating from green-yellow hues to dirty purple and dark brown areas. This indicates the accumulation of hemoglobin breakdown products and tissue compression by gases released by bacteria. Over a period of 1 to 4 weeks, the body influences its environment by releasing organic matter and minerals. Tissues become increasingly softer and looser, and sometimes the body mass loses its recognizable shape and structure. During this time, the role of collagen and other proteins of the extracellular matrix is particularly noticeable, as they lose their properties under the influence of lysosomal enzymes and bacteria. At the same time, the first signs appear that the introduced parasites and insects are controlling the early stages of decomposition. Their life cycle, based on rapid larval stages, is visually evident on the body as clusters of small moving organisms. Their effectiveness is astonishing. They can completely cleanse soft tissue in a matter of days under favorable conditions. One of the most important aspects of this stage is the formation of cadaveric fluid, consisting of a mixture of decomposed cells, bacterial metabolites, enzymes, and decay products. This fluid accumulates in body cavities, causing tissue softening and the formation of putrefactive exudate, which can eventually leak out through the body's natural orifices. The development of these biochemical and biological processes is accompanied by dramatic changes in the body's appearance. Swelling, changes in skin color and consistency, and the odor of decomposition all influence the perception of the death process and often evoke fear and rejection in observers. However, from a scientific perspective, these changes are merely signs of the inevitable transformation of living matter. The weeks and months following death are characterized by increasing decomposition and the loss of the body's anatomical individuality. The skin loses elasticity, collagen fibers disintegrate, and muscles and internal organs become mushy. This stage is crucial for forensic examinations, where the nature of decomposition allows one to accurately determine the time and conditions of death. Natural conditions can radically alter the course of decomposition. A dry climate favors mummification, in which skin and soft tissues dry out and are preserved in an altered form. Wet and anaerobic conditions promote the formation of adipocere, or cadaveric wax, a hard substance capable of preserving tissue shape for years and even decades. The formation of adipocere is a rare but extremely fascinating process. Under the influence of anaerobic bacteria, subcutaneous fats are converted into a waxy substance that prevents further decomposition. This allows the external contours of the body to be preserved, including facial features, ear shape, and even skin texture. Such bodies are often found in sealed coffins or damp areas where oxygen is limited. In contrast, mummification involves intense evaporation of moisture and gradual tissue drying. The skin becomes dense, hard, and characterized by cracks. This process preserves the body, preventing bacteria and insects from actively interfering with its structure. In some cases, mummification can last for decades, preserving a significant portion of the anatomy and appearance. Studying these changes helps us better understand how environmental factors, microflora, and biochemistry interact to create the unique post-mortem state of each body. Understanding the role of microbial expansion and environmental conditions allows us not only to reconstruct the death process, but also to develop methods for preserving, studying, and identifying remains. A detailed understanding of microbial penetration, enzymatic degradation, and insect activity is crucial for forensic scientists. This helps establish the precise time and conditions of death, as well as identify the causes of death and possible signs of violence or disease. This entire picture is completed in the third part of the study, which examines the long-term preservation and transformation of body tissue and bone. Moving on to long-term changes, we see how processes initiated by microbial invaders and insects continue to evolve over months, years, and decades, altering the body's physical appearance and leaving unique forensic traces. Long-term transformations and final destruction of the skeleton. Several months after cardiac arrest, the body enters a new stage of complex biochemical transformations. By this time, the vascular system, once a vibrant network, begins to completely deteriorate. The vessel walls, composed of thin layers of endothelium and connective tissue, become loose and permeable under the influence of bacterial enzymes and decay products. In this state, they are no longer able to retain the liquid components of the blood, leading to further exudate leakage into surrounding tissues, increasing swelling and decomposition. Hemoglobin, released from destroyed red blood cells, begins to oxidize, giving the tissues bizarre shades ranging from brownish to purple. These visual changes become especially noticeable on the skin of the face, neck, and chest, forming a marbled pattern that indicates continued biochemical breakdown. Under a microscope, the tissue structure at this stage is almost unrecognizable. Cell nuclei are destroyed, and the cytoplasm transforms into a heterogeneous mass. Oxidative processes not only change color, but also affect the chemical composition of the tissue, stimulating the formation of toxic compounds. This point is considered a turning point. The body can no longer be revived, and the tissue changes are irreversible. Depending on external conditions, humidity and temperature, the body may begin to mummify or transform into a wax-like state. Mummification accelerates in dry, well-ventilated areas, transforming the skin into a dense, parchment-like covering that resists further deterioration. At the same time, High humidity and a lack of oxygen promote the formation of adipocia, known as cadaveric wax, which preserves the tissue's shape and prevents its decay. Adipocia is a dense mass formed from hydrolyzed fats and fatty acid salts. This unique preservative can preserve the contours of the face and limbs for many years, sometimes allowing even the basic features of the deceased to be determined. This process occurs when the body is buried in a sealed, humid environment, such as flooded graves or peat bogs. It's important to understand that adipocia creates unique conditions, inhibiting microbial activity and supporting nervous tissue. Although these conditions cannot fully restore function. The opposite scenario, mummification, occurs under the influence of high temperatures and constant air circulation. In this case, the skin dries and hardens, while the internal organs partially retain their shape despite the loss of moisture. As the body undergoes these transformations, soft tissues increasingly decompose, oscillating between enzymatic and microbial degradation. The risk of external influences also increases. Insects, fungi, and bacteria continue to actively decompose organic material. Humidity, soil acidity, and the geochemical characteristics of the area influence the rate of decomposition, creating a wide range of variations in the process. Some remains can remain in the adipocia state for decades, others turn into marble mummies, and still others quickly disappear, giving way to mineralized remains. The skeletal system remaining after the death of soft tissue retains its shape. But it too is not eternal. Its strength is due to a strong organic collagen base and a mineral matrix, primarily hydroxyapatite. After several decades, collagen begins to degrade under the influence of soil microorganisms and physicochemical factors. The collagen fibers that give bone elasticity and strength gradually disappear, transforming into a weak, inorganic structure. As Dr. Sergei Volkov, a forensic scientist at the Institute of Forensic Medicine, noted, after 40 or 50 years, the skeleton becomes fragile, easily broken, and visually resembles ancient fossils. This fragility is especially noticeable during exhumation. Bone destruction is uneven. The thin ribs with their hollow interiors and the bones of the hands and feet are the first to disintegrate. These structures lack strength and quickly disintegrate under the influence of external factors. The skull, although quite strong, remains vulnerable at the periphery, especially around the nose and ears. More massive bones, such as the femurs and humeri, may survive longer, although they are subject to deformation. The clavicles and scapulae, despite their flat surfaces, are less stable due to their spongy structure. This fact allows experts to determine the duration of preservation of remains and the conditions of their burial. Teeth deserve special attention. They are composed primarily of minerals, and their enamel is extremely durable, harder than even granite. This allows teeth to last for hundreds of years and is used in forensic medical examinations for identification. However, over time the supporting tissue and alveolar bone deteriorate, leading to loosening and tooth loss. Metal fillings and crowns are often retained, serving as additional markers. Dr. Volkov emphasizes that teeth are a kind of biological archive, preserving the imprint of life for the longest time during the process of decomposition. Environmental conditions have a significant impact on the timing and nature of bone deterioration. Wet, acidic soils, such as peat bogs, accelerate the leaching of minerals, reducing strength and decomposition rates to an average of 30 to 40 years. Alkaline soils, enriched with calcium and other mineral salts, on the other hand, promote bone preservation for up to 100 years or more. A dry, low-temperature climate can lead to partial mummification, the preservation of tissue at the molecular level. Burial depth affects oxygen availability, moisture, and microbial activity. The deeper and more hermetically sealed the burial, the slower the last organic resources are depleted. Decades after death, bodies often turn into dark brown masses and powders. Even strong bones become porous and crumble over the years. As renowned anthropologist Elena Mironova notes, when we work with remains over 70 years old, their structure resembles loose dust, where it's difficult to distinguish individual fragments. This is a natural stage, the transition from biological matter to abiotic forms. After 100 years, most organic components completely disappear. Collagen is completely destroyed. The mineral framework retains its shape for a long time, but gradually deteriorates under the influence of soil factors. In addition to bones, remnants of hair, skin, and soft tissue are lost. Small fragments remain in the form of dust, sometimes combined with soil particles. Forensic and archaeological evidence shows that after a century, minimal traces of personality remain, and DNA, if preserved, is extremely fragmented. The fate of clothing is no less interesting. Natural fabrics such as cotton and linen undergo rapid hydrolysis and enzymatic degradation. After just a few years, only individual fibers or stains in the soil remain. Synthetic materials, on the other hand, are more resilient. Polyester, nylon, and spandex can survive for decades and even centuries, continuing to remind us of their former owner. The memory of human life in such artifacts miraculously outlasts the biological matter itself. 10 years after the body's death, cadaveric wax, adiposa, often forms. It forms in conditions of high humidity and lack of oxygen, such as in dense, sealed coffins or marshy soils. This waxy material is a complex mixture of fats combined with salts, which has antiseptic properties and prevents further decomposition. It preserves the body's shape, including even facial features and muscle contours. The uniqueness of adipocire lies not only in its chemistry, but also in its role as a natural preservative, capable of preserving components for scientific study decades later. The opposite process, natural mummification, relies on the rapid drying of tissues in a dry, warm climate. At low humidity, microorganisms become inactive, and tissues retain their elasticity and structure for a long time. Mummified remains often become a source of valuable data for archaeologists and historians. Absence of moisture prevents the formation of adipocire and radical changes in the structure of soft tissues. The process of bone deterioration continues alongside these changes, critically impacting the condition of remains decades after death. After 50 years, the collagen matrix of bones has virtually disappeared. Remains become friable and brittle, complicating careful assessment and identification. Over the next few decades, bone mass is further degraded by fungi, bacteria, and physical factors in the soil environment. Over the longer term, up to a century, bones continue to disintegrate into smaller particles, combining with mineral elements in the soil. Decalcification and hydrolysis processes occur, which, under the influence of acidity and temperature, alter the structure of the minerals. At this stage, it is impossible to restore the macroscopic shape of the bone, and the newly obtained fragments are powders of varying colors and consistencies. The skull, traditionally considered one of the most durable elements, is not immune to these processes. Its structure becomes porous, brittle, and fragments. Dental enamel, despite its high strength, also gradually erodes, especially in moist soil. This complicates the identification of remains. However, even in these conditions, the discovery of teeth and metal fillings continues to serve as key identifying clues. Thus, the prolonged transformations in the human body after death lead to the gradual disappearance of the physiological structure and the creation of new forms of material existence based on the interaction of biological, chemical, and physical processes. Everything that once constituted a living entity dissolves, transforms, and becomes part of natural biogeochemical cycles. This final phase completes the long journey from life to the eternal return of bodily elements to the environment. Thus, long-term deterioration of the body is a complex, multi-stage process, including tissue oxidation, vascular breakdown, mummification or adipocere formation, and the gradual degradation of the skeletal system under the influence of external and internal factors. Understanding these processes is important not only for forensic science and anthropology, but also for understanding the biological cycle. The next section will explore how nature completes this cycle, transforming the remains of our bodies into soil elements and opening a new chapter in the endless history of matter.